The joint research team led by Wang at Zhejiang University has submitted a review article to iMeta entitled, Metal-Dependent Regulated Cell Death, Molecular Architecture and Translational Frontiers. Overview of the review. Intracellular metal dyshomeostasis drives diverse regulated cell death programs, including ferroptosis and cuproptosis. This review delineates the molecular pathways underlying these death modalities and explores bidirectional translational strategies for targeted tumor eradication and protection of vulnerable tissues. The key highlights of this article mainly include the following 4 points, including revealing that metal dyshomeostasis drives diverse RCD programs. In the introduction, we highlight a paradigm shift in the understanding of cellular metal toxicity, moving from accidental necrosis to precise regulation. Over the past 2 decades, the field of metal-dependent RCD has witnessed exponential growth. From the elucidation of key ferroptosis proteins to the successive emergence of cuproptosis and calcicoptosis, these milestones are continuously redefining the frontiers of precision medicine. Chapter 3 focuses on the core mechanisms of ferroptosis. The left panel of Figure 2 illustrates iron uptake, storage, release, and the disruption of iron homeostasis, while the right panel integrates membrane lipid remodeling, ultimately leading to ferroptotic cell death. Figure 3 presents multiple antioxidant pathways that collectively establish a tightly organized, multidimensional intracellular defense network. In addition, multilayered regulatory switches precisely tune the threshold between cell survival and death. Unlike ferroptosis, Chapter 4 reveals that cuproptosis directly targets the cellular bioenergetic hub. Figure 5a shows that copper entry into mitochondria induces the aggregation of lipoylated proteins, triggering lethal proteotoxic stress and metabolic collapse. Figure 5b further illustrates how glycolysis, hypoxia, and lactate determine tumor sensitivity to copper. Figure 6 reveals the lethal crosstalk among copper metabolism, iron metabolism, and lipid peroxidation. Chapter 5 focuses on several other emerging metal-driven lethal modalities. Figure 7 illustrates how sustained mitochondrial calcium overload forces the opening of the mitochondrial permeability transition pore, leading to collapse of the cellular bioenergetic machinery and triggering irreversible calcicoptosis. In addition, this review proposes the concept of zincyptosis for the first time. When large amounts of label Zn are released and flow into mitochondria, they not only disrupt Fe-S clusters and paralyze mitochondrial metabolism to trigger zincyptosis, but also concurrently unleash ferroptosis and pyroptosis. Figure 9 focuses on noptosis. Massive Mn release within the tumor microenvironment can trigger noptosis while potently activating anti-tumor immunity, thereby unleashing a systemic anti-tumor immune response. Moreover, metal-triggered cell death does not occur in isolation. In the upper panel of Figure 10, nanomedicines release metal ions to unleash a full-spectrum panoptotic storm and cascading lethal effects. In Chapter 6, we comprehensively discuss how foundational mechanisms can be translated into clinical therapeutic strategies. Figure 11 delineates bidirectional translational strategies in the context of ferroptosis, encompassing targeted tumor eradication and protection of vulnerable tissues. Chapter 7 demonstrates how bioresponsive nanomedicines can overcome the toxicity bottleneck of metal-based therapeutics. It further highlights the 4 smart delivery strategies illustrated in Figure 12. Chapter 8 comprehensively introduces systemic immune remodeling in metalloimmunotherapy, mainly encompassing the 3 aspects outlined above. This chapter comprehensively discusses the challenges and future perspectives of clinical translation, focusing on the above aspects. Finally, this review provides a comprehensive synthesis of the network mechanisms and translational strategies underlying metal-dependent RCD. Building on this framework, we innovatively propose an interdisciplinary theoretical blueprint encompassing ferrology, syncology, and cuprology, thereby advancing element science toward an era of precision medicine.